Okay, it might seem a bit of overkill to have two UniProt speakers one after another, but actually I'm not going to talk about what we do, but more about what I would hope to invite you all to get involved in. So Yannis gave a wonderful introduction of our experience with manual curation and automatic curation over the last 20 odd years in my case and 30 years in the case of SwissProt. And we've learned a lot. And one thing we've learned very clearly is that we need lots and lots of people to face the challenge coming from very different areas to build a community for genome and proteome annotation. We're at a critical stage right now, and there's awareness of it. So we organized a workshop in Washington in March this year. So this is a very different kind of talk. It's not about the science, it's more about a workshop report and then asking you to get involved. So we had this meeting at Georgetown University. And I think it's very important to understand that this was actually funded by the NIH through the UniProt grant. When we said we need to work with the community around the world, not just, well, this solution is not around the world, but that's the plan. But UniProt can't solve everything by ourselves. So the idea was to discuss a shared vision for the future of annotation in the genome era, with, of course, because we were organizing it, a special focus on protein functional predictions. And the idea was to have a really good discussion about what we all do and what we would like to achieve and how to build that community. So just to give you an idea of the participants, we brought, because this is based in the States, most of the participants were, if not nationally from the States, they were actually based in the States. But it represented sequencing institutes, some computational researchers, and database resources like RefSeq and UniProt, and some experimentalists as well. And this was just a kickoff. This is a small slide. We just wanted to get together and see what can we do. Is this the right time to start talking about such building a community? So most of you I saw the hands going up earlier. You're involved in CAFA. You know what the situation is. We have loads and loads of genomes. There have been huge advances in genome sequencing technology. As Yannis just said, when we started out in SwissProt, there were no genes. I sat with a paper. It was biochemistry. Yay. Then I had to learn molecular biology. Yay, cool. Then the genomes arrived. Then statistics arrived. We're all evolving. This is great, because I hate to be bored. But you know, I need some help here. And this is the important thing that we've now, I think, come to terms with, which is, yes, we have lots of sequences, but sequences do not equal function. The other thing that obviously my group is passionate about is that there's a lot more to a protein than just giving it a name. There's been a lot of focus in the genome community with saying, this is the name of the protein, therefore you know what it does. We've just had this question about context. It can be the same protein name, but if it's somewhere else, doing something else in a different environment, it's a completely different protein. So in a way, a lot of the genome annotation is quite misleading, and it's a real serious concern. And this is an important point to get this right, because now with the next-gen sequencing, with sequencing moving into labs, people can do it any which way they want. It's really important time to reevaluate where we are. So we had lightning talks from the participants about, we've set them a challenge. What do you do? Short. What do you think are the challenges? And then we broke into groups to brainstorm on our vision of the future of annotation. What are the barriers? What are the solutions? This was actually a great deal of fun. I think Ido said it was the best meeting he had been to. There was a lot of chocolate, I must say. I smuggled it from Europe, both Italian and Swiss, just to be sure. I'm Irish myself, but I can't say anything about our chocolate. The beer is good, but I couldn't bring that to America. And so we had a really successful, I really enjoyed it, and I think everyone who came for the day did. But I'd like to now show you the solutions, and we'd like your feedback on this, of what we think we need to progress in this field. The first of all is establishing the community, but who do we actually want in this community? We need the experimentalists and the experts in annotation pipelines. We need the database providers like me and the curators in my team who work on this all day, and in other teams of UniProt. We need standard developers, this was just raised. and computational researchers, effectively you. As all of them, all of the different expertise is critical for successfully and accurately annotating genomes and proteomes. And that's the goal, to establish such a forum. But it's also really important, and I really like the questions from the previous talk, which is we need to define what it is we're actually trying to do with annotation. So you have the annotation that you find in SwissProt, where a curator sits and writes that. Traditionally, that was all free text-based. The curator read the paper, put it in their head, it came out in English. Varying qualities of standards of English sometimes. That's improved over time. But now we're learning about machine parsable stuff, and we're putting in the control vocabularies, we're putting in the ontologies, we're refactoring how we actually annotate. So in the next 4 years, we're doing a complete data review of SwissProt, where every single type of annotation is being reviewed with the intention of it being both useful for biologists, which it already is, but even more for computational biologists. Any feedback, any questions you have about that, do let me know. But we have actually now, we're very proud, we've only just started with this concept of standard operating procedures. So we now have some on our website telling you how we actually annotate and how we do our automatic annotation. And this is critical not just for us, but also for the annotation pipelines. Because we've been working on importing nucleotide data from resources around the world for a very long time, sitting there and trying to understand how you actually predicted a protein or predicted a function when there's no evidence attribution provided, there's no method provided. The curators are second guessing, and I'm sure, and our users are delightful, but they believe everything we have in our database, whether it's reviewed or unreviewed, is reviewed. So they think a curator has looked at that, when in fact we've imported it from another resource. That's where evidence attribution is critical, and we now have that in UniProt, and hope you are using it. So, as I said, it's imperative for the community to move away from the protein name just as the primary piece of annotation. It's just simply insufficient to actually know what a protein is doing in a particular organism. The other thing that's really important, and this was also raised in the previous talk, is that you need to have a central resource of curated and experimental data. UniProt does a great job. I'm not going to knock the last 20 years of my life and everyone else's effort. But there's things that you need. We're working on improving, but there's other attributes that are needed which are outside the scope of UniProt. So we need to bring all of that information together, and in a way that then the computational researchers can use it. Rather than people taking our data, not always understanding it, and putting it together with data from other resources, which is redundant, and thinking it's actually supporting each other, when in fact it's actually the same piece of annotation with the same evidence attribution, but you don't know it. And we need to understand how that works to ensure that you have proper validation and generalization, that you feel safe to do that with a computational method. So you're making the right inferences and conclusions, and we really want to help with that. The other thing that came up very much in discussion, because there were people there who wrote pipelines and who did write code, and as Yanis mentioned already, people get their PhDs, move on in their careers, and leave what can be a beautiful, beautiful piece of code just sitting somewhere, and no one knows how to use it, and no one knows how to reproduce it. The idea of this forum will be that we will be able to bring all of that together in a central place and establish it in like a series of modules or a pipeline that you can come and pick and use as you like, but also to enable people from smaller resources or individual researchers to come and do that as well. So not only the computational researchers submitting their programs to be maintained when you move on, but also that people can come and use it when they want to send up an annotation pipeline. There's a huge amount of expertise, but there's always, as we learned from this meeting where we had people working in bacteria, people working in viruses, people working on eukaryotes, there's things you need to know when you start off with a different taxonomic range. There's things you need to know about the signatures and their hit rate and what you can tell about them. And all of the expertise, it's out there, but it's not somewhere where you can easily access it. So the other issue, and again, this is a good place for it, is the text mining. You would think as a curator for 20 years, I would be afraid of machines replacing me. Nah. But in fact, I would appreciate if the machines could help me find the most important papers in the best, most efficient way. It's not there yet. It's improving. I've been watching it improve. And this is one of the reasons why we got involved in CAFA and why we're going to be working on the next project to help with this. We really want text mining to work, and we're willing to put in the effort on the validation with that. Happily, we've now got some funding for that, which is nice. But the key issue is that the only way for you to do this properly is if the data is structured in the first place in the papers. If you've ever sat and read papers, it's also incredibly frustrating for curators, because it's still amazing in this day and age that people can leave out the name of the species, never mind the strain, and don't make clear in the experimental methods where the experiment was actually done. So it's a particular skill in learning how to read a paper. It would be much easier if it's for us as well as for programmers if this was structured. We all know that authors, this is a very polite way of saying things, are best positioned and motivated at the point of submission to do this work, as opposed to after. So therefore, it's really important to get the journals in place for this. And the EBI is doing some outreach work through the European PMC to try and achieve this, but there's still an awful lot more to do. It is also really important, and I think any of us who have been involved in annotation in any way, shape or form understand that right now, as Jana said earlier, funding agencies seem to think that all annotation should be automatic. But actually, when you start talking to people, there's a huge misunderstanding about what annotation can be. It can be something as simple to me as gene calling. You've run a program. But then it can be something as developing a controlled vocabulary for function, which you've actually even raised. That's a huge challenge. Even with the gene ontology, it's still not sorted. And then there's the things that we do, which is capturing and propagating functional annotation. And Yannis gave a lovely example with the Proteome family of just how complicated that is. And we have an ongoing joke within the SysProd annotation teams, which is there's always an exception. And trying to explain that to the programmers is just amazing. I have this joy on a regular basis. And this absolute blank look when they first arrive at the EBI, and I'm like, this is just the way it is, okay? They get it by the time they leave. We train them well. But and then the key issue that we all want to do is discovering the function of uncharacterized genes and proteins. So there's still a lot of information out there in the literature that we haven't captured yet, but there's also stuff that just is not known. And we need to build the collaborations to put all of this together and then teach, enlighten the funding agencies about this. So then, this is the thing that's very close to my heart and was also close to Amos's, which is one reason why he left us to go to the human proteome. He also got his own lab. If you're sitting there as a curator, if you're sitting there working in automatic annotation, you know what you don't know. The known unknowns, I believe a certain American president used that. But I'm using it in a much better context, which is basically, I know what we don't know about the sequences in Tremble. And I want to find out, but I need people to go back to the lab or go to the lab and figure it out for me, because I can't. I'm sitting in an office, in a building full of offices, and that's just the way it is. So our experience, there is this disconnect, but when we go to experimental conferences, people really like to work with us, but it doesn't inform the funding mentality, and they don't look in Unipart to see what is not known. They look to see what we have said. We need more data, and I think that applies to all of us. And then finally, so engaging with the cutting edge computational scientists. That's you lot, I hope. And as I said, hard coding the methods identified as useful for our community. So the stuff that you produce, then we can use it consistently, reproducibly, and ongoing for proteome and genome annotation. So please do get involved. I'm here for the entire meeting, and also Ido is involved in this effort. And we're trying to organize another meeting for early next year. Thank you very much.